I'd like to take the time to thank you for giving me this opportunity to share with you my research today. Like Dr. Tots has told you, my name is Leslie Bailey, and I'm from Dr. Wang's lab, and my research is on targeting Lentivectors to CD34 positive hematopoietic stem cells for gene therapy. So I'll give you a brief overview of my presentation, some of the questions I want to answer for you, and some of the topics I'd like to discuss. So I want to answer, what is targeted gene delivery? What are the benefits of targeted gene therapy? And why did we choose to target the hematopoietic stem cell, also known as the common blood progenitor cell? I want to share with you our novel method for targeted gene delivery, our proposed virus entry mechanism, the results that we've achieved, and some of the future work that we plan on doing. So I'm sure most of you have heard of the Human Genome Project. It was completed in 2003, and they sequenced 3 billion base pairs of DNA that include around 20,000 to 25,000 different genes. The central dogma of biology is that you take DNA and you get RNA, and from RNA you can get protein, which is essentially the building block of life. So now that we know the sequences of the DNAs, and scientists are working very hard to study what all the different genes are, this will be a huge benefit to gene therapy. So targeted gene therapy. Gene therapy can be used to augment gene expression level or even introduce a new therapeutic gene. Why would we want to target? Well, if you can target, you can actually do the gene therapy in vivo. You can avoid off-target side effects, for example, introducing the gene into a cell that you don't want to introduce it into. You can also increase the probability of delivering your gene to a specific cell. And this is really important for my case. So I'm trying to target the human hematopoietic stem cell. And there's only about 1 in 15,000 cells are hematopoietic stem cells in the bone marrow. And only 1 in 100,000 cells are hematopoietic stem cells in the peripheral blood. So as you can imagine, if you're not using a targeting mechanism, it would be very difficult to deliver this gene in vivo. So to target the hematopoietic stem cell, we looked at what markers were on the stem cell. So CD34 is the only reliable marker for hematopoietic stem cells. So we chose to target CD34 using an anti-CD34 antibody. So why are we targeting hematopoietic stem cells? Well, one of the reasons is that it can generate itself. It can self-renew, and it lasts the lifetime of a human. So you can have a lifelong supply of your therapeutic gene if you're able to introduce it into the hematopoietic stem cell. Also, the hematopoietic stem cell makes all of the cells in your blood, including very important cells for your immune system, including T cells and B cells. NIH has a list of possible diseases that you can cure and problems that you can fix with the gene delivery to hematopoietic stem cells. These include boosting your immune system to cure diseases that we don't have vaccines for yet, such as cancer or HIV. You can also cure inherited blood disorders, like some types of anemia or severe combined immune deficiency disorder, among others. And you can also use it to cure inborn errors of metabolism. So there's really a large potential for gene delivery. The way we plan on delivering our gene is using a lentiviral vector. So viruses are highly evolved gene delivering machines. So we can use this lentiviral vector, which integrates the gene that we put into the host cell genome. So the gene will be expressed the life of the cell. It can transduce non-dividing cells, so it's a very powerful tool. And since hematopoietic stem cells don't divide very often, this is very important for us. It's an enveloped RNA virus, and it's derived from HIV. So I have a brief description of the vector that we're using right here. And we call it FUW. It has a ubiquitin promoter, so it can promote in pretty much all mammalian cells. You can introduce any gene of interest that you would like in the middle, and it has a woodchuck responsive element to increase the half-life of the RNA. And it's been engineered so that the safety level is increased, and I can talk more about that if you have questions about that later. The current method of gene delivery to hematopoietic stem cells involves bone marrow transplant. It's very tedious, and it's a personal medicine. You have to do it especially for each person. You can't just transfer any bone marrow into any patient. So before the stem cell transplant, they have to mobilize the stem cells. And then they have to collect the blood cells. They select the blood cells using CD34 selection, which should include some of the hematopoietic stem cells. Then they are able to finally infect the hematopoietic stem cells, and they have to freeze them until the patient is ready to receive them. To make the patient ready to receive them, they have to irradiate the patient, or they need to use chemotherapy drugs to prepare the patient for receiving these stem cells. And finally, the patient can receive the stem cells. It doesn't always work, it's variable, it's high cost, and it's tedious. So this is not a possible third world country cure. You couldn't use this for HIV in Africa. This would be a very difficult problem. So our method is kind of it will be a novel type of vaccine. This vaccine you could use like any other vaccine. You would deliver your virus, which is going to have the anti-CD34 antibody, and then your gene of interest, which could be to help cure the disease or to replace a gene that somebody's missing. And you could use this widespread. It would be much less expensive. It's actually a possible method of in vaccinating people in third world countries even. Our virus would ideally find the stem cell and deliver its therapeutic gene in vivo. So the current methods of targeted gene delivery, they use the one glycoprotein on the cell, on the virus surface, and try to incorporate targeting into it. And this really decreases the titer and infectivity of the virus. So it doesn't make it useful for therapeutic purposes. It's not high enough titer. People have also tried to bridge an antibody between the virus and the target cell. And that is really low titer in vivo, and it just isn't going to be a high enough titer. So our method, we looked at what were the two important things that were required for entering the virus into the cell. And they are binding and fusion. So we decided we could separate the binding and fusion functions so that we won't have to sacrifice the fusion for the binding, and we won't have to sacrifice the specificity of our virus for infecting the other cells. So to separate binding and fusion, we use the natural budding mechanism of the lentivirus. It actually incorporates the cell membrane onto its outer layer when it exits the cell. So we can just express on our cell that we're using to make the virus, we can express the antibody and the fusogen, and our lentivirus that will have our gene inside of it will express both our antibody and fusogen as it buds out from the cell. This allows for therapeutic titers, and this method is actually quite broad. You can use it to target antibodies, antigens, ligands, or receptors. So really you could use it to target all kinds of different cells, not just hematopoietic stem cells. So how do we propose that our virus actually enters the cell and works? Well, our virus will bind to the hematopoietic stem cell via the CD34 receptor, as I described before, and this will cause endocytosis. So one of the important things that when you consider what you're going to put on your virus is that it needs to cause endocytosis when it binds to the cell. As it is endocytosed, when it's in the endosomal compartment, the pH lowers, and this triggers fusion. So the fusogen needs to be pH specific. When the fusion is triggered, the viral capsid is released because the virus binds to the endosomal compartment. So looking at the two different necessary things that we were putting on our virus, we wanted to make sure they were necessary and that they were really working. So first we looked to see if there's actually binding going on. So we took a cell line that we made. We took human embryonic kidney epithelial cell, and we put CD34 on it. We made a stable cell line. And then we took our virus that had anti-CD34 and the fusion molecule, and then a virus that doesn't have the antibody on it, just the fusion molecule. And we used FACS assay to analyze it. So we expect if the virus binds to the cell, it won't be washed away, and you can stain for the virus and the cell and see if they're both together in the same location. However, if the virus doesn't bind to the cell, it will be washed away, and then you won't see it in the FACS assay. So you expect a shift if there is a virus. So you can see the shift here. So we did have binding between our virus and the cell. You don't see the shift when there is no antibody on the virus, just as we had hoped and expected. Then we wanted to make sure that this binding was really necessary. So we decided to add soluble CD34 antibody and see if that would compete with our virus to bind to the cell. And so we predicted that if we add, the more CD34 antibody that we add, the less transduction we're going to get, or the less infection we're going to get. And as you can see, that's exactly what we got. The more CD34 antibody we added, the less infection we had. So binding is absolutely necessary. Then we wanted to look at fusion, the second important molecule that we're putting on our virus. So first we wanted to see if we could see fusion. So we've got one cell that has the fusogen on it, and it'll bind to another cell here that doesn't have the fusogen on it, but it should bind. And this cell is not GFP positive, but this cell is GFP positive, and GFP is green fluorescent protein. And then we lowered the pH to see if our cells would bind together. So what we expected to see would be a multi-nucleus mass of green cells if they actually do bind together. So you can see here at the pH of 7.4, the cells are all separate and there's not a multi-nucleus mass. But when we lower the pH to 6.0, like it would be in the endosomal compartment where our virus would fuse, you can see these multi-nucleus masses of cells. The nucleuses here are going to be the black circles. So we were able to see fusion. Then we wanted to make sure that fusion was absolutely necessary for infection. So we predicted that if we increased the pH in the cell, that our virus wouldn't be able to work because the pH would be too high and the fusion, the endosomal compartment couldn't reach the low pH that it needs for the virus to get released from the endosome. So as we predicted, as we drop the pH using ammonium chloride, we see a steep drop in infectivity of our virus. So now we want to see really how good is our virus. Is it really targeting specifically, or are we going to get a lot of background targeting? So to do this, we have two cells. The only difference is that one cell has CD34 on it, which is the marker we're trying to target. So we used our virus to target both this CD34 negative cell and the CD34 positive cell. And we hope to see that the CD34 positive cell is getting infected and not the CD34 negative cell. So here we used FACS analysis again, and we're looking for green fluorescent protein, which is what we're delivering with our virus. And you can see the filled section is targeting infecting the CD34 negative virus, and the open section is infecting the CD34 positive virus. If you see a shift in GFP, that is infection. So you can see that our open section is shifting for these different fusion molecules. And you can see really it shifts the most for fusion molecule 3, so that's the most efficient fusion molecule we've found so far. Doesn't seem to be infecting much at all with fusion molecule 4. So picking the fusion molecule is very important. But definitely we have specific targeted infection in vitro. So the real next step is to see if we can do this in vivo. So we set up a mouse model. And in our mouse, we put CD34 negative cells on the left side of the mouse, in the left flank. And we put CD34 positive cells on the right side. We use our virus this time to deliver firefly luciferase so that we can add a substrate, and you can actually keep the mouse alive, and you can inject the substrate and use this imaging technique to see if there's any firefly luciferase being expressed. So on day 7, when we injected the virus, we began to image our mice to see if it was glowing at all. And we expected that if we had specific targeting, it would only glow on the CD34 positive side, which will be the right side of our mouse. And it shouldn't glow on the CD34 negative side. So as you can see here in our mouse, right here, this is our targeted mouse, it did only light up on the targeted side. So this is very exciting. For our non-specific targeting, this type of coating on the virus, the SVG, will infect most mammalian cells. It lights up on both sides, so it's not targeting. And our no virus control doesn't light up at all. So we definitely have specific targeting in vivo, which is very exciting for me. So for our future work, we want to move this into primary cells, primary hematopoietic stem cells. And we hope to see primary cells targeted in a mouse model also. And eventually we'll move this to clinical therapies. And we can try to, like I mentioned, boost the immune system to cure cancer or HIV, cure inherited blood disorders, or cure inborn errors of metabolism. Our lab is also working on improving the fusion molecule to see if we can get a better fusogen and even increase the titer of our virus. I'd like to acknowledge Dr. Wang's lab, and especially Dr. Wang. He's been an excellent advisor, and he really helped me get me started and train me on doing this work. I also would specifically like to thank from our lab, Tae-hoon, Alex, Eric, and Wang. They've all helped me a lot. They've helped me with cloning and making virus, and the whole lab's been very supportive. I'd like to thank our collaborators. The Baltimore lab has kind of taken me in. I've gotten to do a lot of my work. They've really made me feel like part of the lab. Professor David Baltimore is always insightful and has lots of great ideas. Lily Yang she's been like a mentor to me. She's really helped train me, and she's very patient with me. And Mandy Wang has helped me do a lot of the in vivo experiments. Professor James Strauss has provided us with some of the materials, and Professor Don Kahn has given us lots of insight into this process. Our funding has come from the Bill and Melinda Gates Foundation, and specifically the Grand Challenges in Global Health and the National Institute of Health. So are there any questions for me?
Thanks, Leslie. Very nice job. Any questions for Leslie?
Yes.
Could you back up your slide a little bit?
Sure.
From there. Again, what technique are you actually using to, I'm seeing images there. What's the technique? Is spectroscopy, ultraviolet light, what
It's actually like a very sensitive camera. It's letting off visible light. It's like firefly. You know, the firefly lights up. It's letting off visible light, and we have a very sensitive camera that's able to detect the amount of visible light that's coming from our mice. So.
Okay, so it's a luminescence or something.
Yeah, it's luminescence, bioluminescence. That's right.
Okay.
Yes.
And just to comment that as a person who has a vascular disease, I find this very personally interesting.
Great, thank you.
Martin?
Where do you do the mouse test, and where are you going next with animal models and in vivo?
So where do we do the mouse test?
Yeah, and then what are you going to be doing next in vivo? What are the plans?
Okay, so we do the mouse test at Caltech. We have an imaging machine that takes the pictures. And the next test is actually a little complicated. Somebody from the Baltimore lab is working on making a humanized mouse model. So they're going to have, like, the blood system will all be like a human. So we can target in the humanized mouse model the CD34 cells. So that would be the next step to target in vivo. We can also try targeting using this scheme, using like a tail vein injection, so it's not directly injected into the tumor, to see if we can really get it throughout the whole mouse and target the tumor that's growing up. Any more questions?
Any more questions? If not, then let's thank Leslie one more time. So our next speaker is Mr. Wei Chen.